Hi everyone, welcome to your online lecture for the course Altered Physiology 1. In today's lecture we're going to be talking about genes and genetic diseases. This is the second lecture on this topic. We're going to look at dominant recessive autosomal and X-linked disorders as the majority of the lecture content, and then we'll briefly cover the concepts of penetrance, expressivity, and multifactorial conditions. As a review, we know that the human cell has 23 chromosome pairs equaling 46, as demonstrated on this karyotype or karyogram. And for each chromosome, you have one from the mother, one from the father. Now, of these 23 chromosomes, 22 are autosomes. So from 1 all the way to 22 are called autosomes. And then the 23rd pair are the sex chromosomes. And this will either be XX for female or XY for male. Now the Y for males is very small and it contains very little genetic information, and so a lot of X-linked disorders, as we will see later, affect males because they do not have protection from the Y. Now we're going to discuss some important terms, and we're going to refer to this zoomed in image of chromosome 7. So you can see chromosome 7 here, and each gene that is part of that chromosome occupies a position along a chromosome, and that is known as a locus. So the genes found at a particular locus can have different forms. So that is, they can be composed of different nucleotide sequences, and we call these alleles. So different forms of a particular gene at a given locus. Now, a nucleotide, as a reminder, is a molecule that consists of a nitrogenous containing base. So remember, for DNA, that was adenine, guanine, thymine, or cytosine, and then in place of thymine was uracil for RNA. And it also consists of a phosphate group and a sugar. So that's just a reminder from normal anatomy. Now, if we look at this chromosome here, one from the mother, one from the father. At a given locus or point, a person will have one allele from the mother and one allele from the father. When these two alleles are identical, we call them homozygous. And when they are different, we call them heterozygous. Now I'm going to give you a different example that's shown here on the screen, but I'll come back to it. So an example of the alleles being the same, homozygous, so this is the same as this in terms of the information, could be that they both code for blue eyes. So this codes for blue eyes and this codes for blue eyes. So they are the same, so they're considered homozygous. An example of them being different or heterozygous could be that one codes for blue eyes and one codes for brown eyes. So they are different. Now the combining of these alleles, so we had an example of blue eyes, or blue eyes, brown eyes. The combining of these alleles can give rise to an offspring's genotype. So now we're jumping to the top of the slide. So the composition of genes then at a given locus is going to be known as the genotype for that offspring. So genotype, right down to the genetic level. Now the outward physical appearance of the individual, which is the result of the genotype, is called the phenotype. So I always thought as PH for what is physically expressed in a person. So what we can say is that the genes of brown or blue eyes that are inherited from the parent are the genotype. So let's say the genotype is going to be for brown, let's say blue eyes from the mom and brown eyes from the dad. But the expression of actually having brown eyes in an individual is the phenotype, that physical characteristic that actually shows. So at the genetic level, there's a combination of blue and brown eyes, but what is expressed in that individual is brown eyes. So let's look at blood type as an example. Somebody could have a genotype of AA, representing blood type A, or blood type A and O. That could be their genotype, one of these two, and they can end up with the phenotype of type A blood. And we'll talk about how that happens shortly. But a person that has type A blood is going to have a phenotype, which is going to be A. And then going back to talking about homozygous and heterozygous, homozygous referring to the same, a person with type A blood is going to have a genotype of AA, so inheriting A from the mother, inheriting A from the father. And that's because we're talking about a situation where it's homozygous, identical genes. In terms of heterozygous, AB would be an example, A from the mother, B from the father. So AB blood type, but it would also be presenting as different genes on a pair of loci. So it could be A here and B here versus homozygous A and A. And polymorphism, a term I haven't brought up yet pertaining to alleles, is just indicating that what we can see is several different forms. So a locus that has two or more alleles that occur often. So there are more than two different eye colors. There are more than two different blood types. Poly means many, so there are many different possible forms. Okay, we know that genes come in pairs. We inherit one chromosome from each parent and the genes along the chromosome. Genes on the chromosome can be considered dominant or recessive. So we're going to look at the difference between those. But as a reminder, an allele that we talked about on the previous slide is a different form of a particular gene. So the allele for brown eyes and the allele for blue eyes, etc. Now the effects of one allele can mask those of another when those two are found together. So in other words, if we have a heterozygous situation where we have two different alleles, one can be masked by the other. For example, the allele for brown eyes, let's say B, and the allele for blue eyes, so this is capital B, is a lowercase b. The genotype would be capital B, small b, because those are the alleles that make up the genotype. And it's heterozygous because they're different. But the phenotype, what actually displays in that person, the physical expression, is going to be brown because brown is considered to be dominant. And it dominates over the smaller, recessive blue allele. So the allele whose effects will be observable then physically are considered to be the ones that are dominant or strong. And so the opposite is true of recessive, where the effects are hidden behind the dominant gene. And so in order for a recessive gene to be actually expressed physically in the person, it needs to be in its homozygous form, where both alleles are the same. Blue eyes, blue eyes. And the reason in this case, if somebody inherits this is their genotype. The reason they would have blue eyes is because I've told you that the lowercase b represents blue eyes, and the other one is also for blue eyes, and there's no dominant or capital B indicating brown to override the recessive one. So the recessive gene is expressed. This is an example here of the genotype being brown eyes, so one from each parent, and both are capital, both are considered dominant, so this will absolutely be expressed physically. And so that's what you'll see consistently throughout the rest of this lecture, and when you learn about genetics, is that the dominant allele is going to be represented by a capital letter, and the recessive allele is going to be represented by a lowercase letter. Now let's look at dominant versus recessive disorders. A person has a dominant disorder by inheriting the disease gene from one parent. There are no carriers for dominant disorders. You either have the condition or you don't. So if I gave you an example here and we just changed the meaning of this, let's say that this capital letter here is representing a disease, and the small letter is representing healthy. Since the diseased gene or the diseased allele is dominant, it's going to overpower the healthy recessive. Diseases that are caused by an autosomal dominant disease, so referring to these first 22, are going to affect both sexes equally. So that's just a rule that if the dominant disease of an autosome occurs, it's going to affect both sexes equally because it's not pertaining to the sex chromosome. We're talking about the autosomes. An example of an autosomal dominant disorder is called Huntington's. And so if you have that disease gene, it will dominate over any healthy one that's present. Recessive disorders. A person will have a recessive disorder if they inherit the recessive gene from both parents. So remember, recessive is representing the small. And so you'd have to inherit the recessive gene from both parents, that disease gene from both parents, in order for it to be expressed. Sickle cell anemia is a good example. So if an offspring inherits the recessive gene from both parents, then it will be expressed. But if they only inherit it from one, then they're going to have one healthy gene and one recessive gene that is diseased. So let's see here. With sickle cell anemia, actually I have an example down here. Because it's a recessive disorder, it's going to be impacting the lowercase letter in our examples. And if, let's say, somebody has this disease gene here, so this lowercase s, but they inherited that from, say, the mother, but from the father, they inherited a healthy s, as healthy allele as part of this genotype, that healthy dominant one will dominate over the small one. And that person will carry this unhealthy allele, but they won't actually demonstrate it. So they won't have the phenotype. Whereas if they inherit the diseased recessive gene relating to sickle cell anemia from the mother and from the father, then they will have the disease. And we'll look at this a lot closer in upcoming slides, which will hopefully help a lot of this make more sense. Okay, recurrence risk. What is recurrence risk? So parents that are at risk for producing a child that might have a genetic disease almost always ask the question, well, what are the chances that our child is going to have this disease that maybe the mother has or the father has, or maybe the parents carry some disease, and they want to know, if we have kids, what is the chance that we'll pass this on? So the probability that an individual will develop a genetic disease is termed recurrence risk. So for example, if you have one parent that's affected by an autosomal dominant disease, so talking about a heterozygous one, and the other one is unaffected, you'll see that there's a 50% risk of that being passed on to the child. And once we look at this closer, that will make a lot more sense. So once you learn more throughout this lecture and then you study and you come back to this slide, it will make a lot more sense. What's important to know about recurrence risk, though, is that each birth is an independent event. In other words, there's no more or less risk for the first or the second or the third child. So if you have your first child and they don't have the disease, some parents would worry, oh, does that mean there's an increased chance that my next will? And that's not true. For every single offspring, the risk is the same. So what we're going to be doing shortly then is applying these concepts, what we've learned about dominant and recessive disorders and recurrence risk, to real life examples. And as I said, that should make things a lot more clear. Okay, we're going to talk for a moment about a pedigree chart. A pedigree chart summarizes family relationships and shows really which members of a family are affected by a genetic disease so that you can see how it might be passed on. And so generally the pedigree will begin with one individual in a family, and this individual is usually the first person that is maybe diagnosed with a particular condition so that they can see what could occur down the road with procreation. There's a term that's relevant when it comes to passing on genetic disorders called consanguinity. When two individuals that are related produce offspring, the recurrent risk, which we defined on the previous slide, of recessive disorders increases dramatically. So this inbreeding is referred to as consanguinity. So it's a consideration when you're looking at a pedigree as well. So on this chart here, it outlines some of the most common symbols that are used in pedigrees. So just to show you some examples, oftentimes a circle represents a normal female, square for normal male. When you have a bar here, a single bar, it indicates that mating has occurred. So you can see that this normal male and this normal female have mated, and then this is representing their children. And you can see normal female child, normal female, and normal male. And it's also showing you generations here at the side. The double bar represents that consanguinity term that I had mentioned, where there's mating between close relatives. So we'll see some of these symbols come up again later. Okay, moving on now to talk about what are called a Punnett square. So what you see here are two examples of a Punnett square, which is just a very simple diagram that's used to determine the probability of an offspring having a particular genotype and the risk that they will actually have a condition. So we're going to learn first how to fill in these charts, and then we're going to be looking at how to come up with recurrence risk, risk of being a carrier, risk of being normal, and whether or not males or females are more affected. So we're going to go through a number of these examples. The first type of disorder that we'll talk about is called a single gene disorder. These are conditions that are caused by one single gene, and we're going to be looking at autosomal dominant. So it's always important to look at your slide headings so you know what we're referring to. Okay, so this means that all it takes is one affected gene from one parent because it's dominant in order for this to be passed on. And so a reminder that when we talk about the autosomes, we mean we're not talking about the sex chromosomes. So we're only looking then at chromosomes 1 through 22. In this situation, we are going to represent, because it's an autosomal dominant disease, we're going to represent the disease gene as capital D, because the disease again is dominant, so we have that capital D. And so the normal one is going to be represented by lowercase d. So how do we come up with the genotype then of a person that has the condition? Well, their genotype would look like one of these two, either capital D small d or capital D capital D. So this capital D would be inherited from, say, the mother, and this would be inherited by the father. And so because the disease gene is a dominant one, it's going to override and overtake or dominate against that healthy recessive one. So even though they have a healthy gene, this one's going to be expressed because it's a dominant condition. Similarly, if somebody inherited the disease dominant disease from the mom and from the dad, then obviously it would also be expressed phenotypically. So if you don't have the condition, the only option here is that you inherit a normal gene from mom and a normal gene from dad. Now, how do we calculate things such as the risk that somebody will have the condition versus carrying a condition? Is there more risk for being male versus female? What's the risk of just being normal? How we can do that is through a Punnett square. So again, that's to determine the probability of an offspring having a particular genotype. So what we would do here is we would take both situations with the parent. So we have an affected parent and an affected parent. And shown below is that affected parent's genotype. So in this case, it doesn't really matter if it's male or female, if this is mother or father. We have capital D, small d, capital D, small d. So this is the genotype for this parent, and this is the genotype for this parent. And so what we would do to fill in the Punnett square is place both either parent on either side, place their genotypes here, and we would fill it in as so. So capital D, capital D, capital D, small d, and then we would do again capital D, small d, and small d, small d. And the reason it's written in this order and not, say, the small d first is just because this is a more common way to display it. So now what we're going to do is, of these 4 possibilities, we're going to find out what are the chances of a offspring inheriting certain genotypes. So starting with recurrence risk, remember recurrence risk is the probability that an individual will develop a disease. So a parent, say you have 2 affected parents that want to know what are the chances that my child will inherit the disease that we have, a Punnett squared can be done to determine that. And remember that the recurrence risk is the same for each child that they have. So recurrence risk, well, let's have a look at how many diseased genotypes there are. So there's 1, 2, 3, and 1 healthy one. So there's going to be a 3 out of 4 chance of recurrence risk, as you can see, because of the dominant gene that's present in these genotypes. So a 75% chance that each child they have will be affected with this disease. What about risk of being a carrier? Well, I told you already that when it comes to a dominant disease, there is no risk of being a carrier because you're either going to have the disease or you're not when it comes to a dominant condition. So it's a 0% chance of being a carrier. What about the risk of being normal? Or we could say chance of being normal. Well, there's only 1 normal genotype here. So there's a 1/4 chance, or 25% chance, which is really the difference from left from the recurrence risk of that child being normal. What about male versus female? Well, you should know if it's an autosomal disorder, it's an equal chance of affecting males versus females. Now let's look at another situation where we have 1 affected parent with the genotype capital D, small d, and we have a normal parent, so they have 2 healthy genes. They want to know, they go to a genetic specialist and they say, look, one of us has this disease, one of us doesn't. What are the chances that our offspring will? So we complete the Punnett squared. We have our D's placed appropriately. So what is the recurrence risk? What is the chance that they will pass on this disease? First, we need to have a look at where we see that disease gene, that capital D, and we see it in 2 occasions. So we have a 2, a 4, or 50% chance that it will be passed on. And so this is an example that I gave you earlier about Huntington's disease, that if you have an affected parent with Huntington's disease and a normal parent, there's going to be a 50% chance that they pass that on to their offspring. And that's a pretty high risk for a very serious disease. We know there's no risk of being a carrier because there's no such thing when it comes to dominant diseases. Risk of being normal is just going to be the difference of recurrence risk, but we can confirm that by finding here that we have 2 normal genotypes. And male versus female is going to be equal. So this goes back to one of the slides that I'll bring up again, where it talked about recurrence risk of an autosomal dominant trait. Hopefully now you understand why, instead of just memorizing it, that there's a 50% chance of passing on that condition if one parent has a dominant disorder. Now let's take what we've learned about autosomal dominant disorders and summarize it in what we call the rules of autosomal dominance. Now, first of all, we know that because it's an autosomal disorder that we're talking about, it's affecting chromosomes 1 through 22 and not the sex chromosome, so both males and females are going to be affected equally. Similarly, their offspring is going to be affected equally as far as being males or females, so there's no sex link. Overall, these types of diseases are considered to be rare. It's very unlikely that if you have an autosomal dominant disease, that you're going to mate with somebody that also has that autosomal dominant disease. The most common occur in fewer than 1 in 500 individuals, and there is no skipping of generations. So let's look at an example with achondroplasia. In this picture here, you can see a cute little girl that has a disease called achondroplasia, and this is an autosomal dominant disorder. It specifically is a bone growth disorder characterized by having short limbs relative to their trunk length. They will have a more prominent forehead, a low nasal root, and redundant leg folds, as you can see in the arms and the legs. So these are considered phenotypes, right? Characteristic phenotypes of the condition achondroplasia. Now we're going to look at this pedigree here. Remember, we spoke about pedigrees earlier, and you can see that we have a male and female at the top here that have mated. Remember that the circle represents female and the square represents male from the symbols we spoke about earlier, and that line represents that they've mated. And the red here is representing the affected female, okay, that has the condition achondroplasia, this dominant disorder. So we have generation 1, 2, 3, and 4 shown in this pedigree. And you can see that some of the offspring in each generation may end up with achondroplasia because we know, as I just said, that it does not skip a generation. So let's have a look. If we have an affected female that mates with a normal male, and they have 6 children, so 1, 2, 3, 4, 5, and then 6, so all with a direct line to mom and dad. 3 of the 6, in this case just happens to be 2 females and a male, have the disease, the autosomal dominant disease, achondroplasia, and 1, 2, 3 do not. And then these children go on to have their own children, which would be the grandchildren of the parents that we spoke about at the beginning. If we look at one of their kids, this male, that mates with a normal female, and they produce 1, 2, 3, 4, 5, 6, 7 children, these people have a lot of kids, you will see that 1, 2, 3 of those children have the condition and the rest do not. And you can also see going forward that it is into the 4th generation. So no skipping generations, and both males and females are affected equally. So the last comment I'd like to make here on this slide is that if neither parents have the trait, like in this example here where this child, or this person rather, mates with this person, neither of them have the condition, then none of their children will get it. Okay, Now we're going to look at single gene disorders, but specifically autosomal recessive. So what we've covered already were single gene disorders, autosomal dominant. Now we're on recessive. Okay, we're still non-sex linked then, right? And we know that recessive means that you need to inherit 2 copies of that non-dominant gene, the recessive one, in order for it to be expressed. And the condition that I mentioned to you earlier is sickle cell anemia. So let's have a look here. We know that the disorders are not located on sex chromosomes. It's 1 through 22 are autosomes. We know that now the normal gene is actually the uppercase letter, and the disease gene is the lowercase letter, and that's because the lowercase is representing recessive. So in order for you to have the condition, your genotype would have to look like this, lowercase d, lowercase d. So you have to inherit it from both parents in order for you to have the disease because it's recessive. Now, to not have the condition, you need to have that dominant, overpowering, normal gene, that capital letter, in order to be considered normal. So the genotype without the condition would be in either of these situations where you either have the uppercase D, which is normal gene, and then you have the disease gene. Let's say this is representing sickle cell anemia as our example. I had it listed as S on a previous slide, but the letters don't really mean anything. So in this case, they have the disease gene, but they also have a healthy dominant gene that overpowers it. So this person would not have any phenotypic characteristics, but they are still carrying that disease gene. And we'll see what that looks like a little bit more shortly. And then, of course, if they had 2 normal genes, then they wouldn't have the condition either. So let's look at some examples. In this case, with our Punnett squared, we have a carrier parent. Okay, so this would be the carrier example. They're not phenotypically, they don't have any physical characteristics that they have the condition, but they do carry it. And why that's important is because they could pass that on. So if you have a carrier parent that mates with somebody who is normal, let's see what the Punnett square would look like. So we'd have it set up like this. And then we would look at first our recurrence risk to see what the chances are that their offspring will have the condition. So we need to look at what's the condition. Well, really, it would be any time we see this. So we don't see that at all. So that's interesting because that tells us that there is a 0% chance in passing on a condition if you are just a carrier mating with somebody who is considered normal. And for the record, when I say normal, I mean they don't have the disease and they are not a carrier. They are neither. So what's the risk of being a carrier? Well, we know that a carrier looks like this. So we're going to say, okay, there's a carrier and there's a carrier. So not a carrier, not a carrier. So we're going to say that there is a 2 out of 4 chance that you will carry it. So why that's important is because even though your children won't have the disease, they're going to be potentially a carrier, which becomes important when they have children. And so risk of being normal, meaning no disease, but also not a carrier, is also 2 out of 4. And then males and females, we know that it affects males and females equally. Now, if we look at a situation where we have a carrier parent mating with a carrier parent, so both of them maybe, let's say, carry the sickle cell diseased recessive gene. Let's see what their Punnett square would look like. So their recurrence risk then, so the chance that the person will have the condition or disease would look like this. So 2 carrier parents then have a 1 in 4 chance of passing the actual disease onto their child. Risk of being a carrier, so we know that that would just be this situation here, which we see twice. So there is a 2 out of 4 chance that their child will be a carrier of the condition. And the risk of being normal now is going to be the difference really between these. So 1 in 4 chance of being normal, this is our only normal situation, and males and females are affected equally. Now let's move on to if you're a carrier, so you carry the sickle cell gene and you mate with someone who actually has sickle cell anemia. Let's look at the Punnett square. So this is where it gets a little bit more tricky. So for procreation, so recurrence risk, the chance that their child will have the condition represented by DD is 50%. So if an affected parent with a condition, an autosomal recessive condition, mates with somebody who's a carrier for that condition, there is a 50% chance or 2 in 4 chance that they will pass that on. Risk of being a carrier then would be the difference shown here, 2 out of 4. But what's interesting is there is no chance that they will be normal, meaning they will either be a carrier or they will either have the condition. And males and female risk being equal. Well, hopefully this is starting to make a little bit more sense as it's all coming together, and it would be a great idea to review your slides in the next couple of days after watching this because more will start to make sense. So what we've done so far is we've looked at autosomal dominant disorders and we talked about some of the rules. We've looked at autosomal recessive disorders using the Punnett square, and now we're going to look at some of the rules. Now, autosomal recessive disorders are considered rare, just like autosomal disorders are, but the number of carriers of a disease can actually be quite high. And sometimes, a lot of times, you don't even know that you're a carrier for a condition unless maybe you have an offspring that develops it and has the clinical manifestations of a condition that warrant testing, or maybe you just found out some other way, but quite high in terms of carriers. Males and females are affected in equal proportions. We know that because it's not a sex-linked condition. We talked about the concept of consanguinity, which is that mating by close relatives and how that increases the risk of a person getting the disease and passing it on. The disease can be seen in siblings, but not their parents. Now, this is because a lot of the time we aren't even aware that the parents were carriers, and then they have kids, and all of a sudden now they have a condition. 25% of the offspring of carriers will be affected, so that's not something you would go and memorize, but rather you would try to understand what we've already spoke about, that if you are a carrier and the person you mate with is a carrier, your risk of developing the condition is 25%. So 2 carrier parents have there's a 25% chance that their child will end up with the actual autosomal recessive disorder. Autosomal recessive disorders are characterized by delayed age of onset, meaning that oftentimes you don't see the issues until later on in life rather than being born with the obvious clinical manifestations. They're also characterized by incomplete penetrance and variable expressivity, which are 2 concepts. So the penetrance and expressivity that will be defined and discussed later on, so you're best to come back to this slide after and apply what you've learned. Another example of an autosomal recessive disorder is cystic fibrosis. Cystic fibrosis is the most common lethal recessive disease in white children. 1 in 2,500 births are affected, but 1 in 25 white births are carriers that are considered to be phenotypically normal, meaning they have 1 copy of the affected gene, so they physically present normal, but they carry the disease, a diseased gene. And so what's sort of scary about these conditions is you can have 2 parents that are carriers of a condition, but they don't know it. and then they have a child, and that child can be normal, or they can have a child that is a carrier, or they can have a child that actually has the disease, that 25% chance we spoke about before. And so when we look at this area up here, we have a normal unaffected homozygous genotype would look like this. A carrier unaffected, meaning phenotypically normal, heterozygous trait genotype would look like this. Okay, that small d is representing the disease, overpowered by the normal gene. And that the affected homozygous, which we know is small d, looking back at our previous slide, is 25%. And then this here, that double line is showing you the increased risk of a recessive disorder when there's consanguinity that occurs. And that's another way to show the Punnett square. Now let's move on to sex-linked disorders. Now, as you know, the sex chromosomes are X and Y, so it's either going to be XX for female or XY for male. I've also told you already at the beginning that the Y chromosome contains very little genetic information compared to the X chromosome, and because of this, most sex-linked traits are found on the X chromosome and are referred to as X-linked. If a male inherits a diseased X chromosome, he will inherit the disease because they don't have another X to mask or balance out that abnormal gene. There are X-linked dominant disorders. Okay, so we're talking about sex-linked, that 23rd chromosome. Or there are also X-linked recessive disorders. The dominant ones are considered to be rare, whereas recessive are considered to be more common. This slide shows us really the rules, some of the common features of X-linked disorders. And as I've already mentioned to you, I suggest you don't just memorize this slide, but rather try to understand some of the details. So what we're going to be going through on the next handful of slides is examples of sex-linked disorders, and then you will see why these are true. So come back to this slide after we've gone through some of our examples next. So first what we're going to look at is X-linked, so a sex-linked of the 23rd chromosome recessive disorder. The capital H is representing normal, and the small h in this example, and the letter is just random, the small h is representing the disease gene. Why is it the small h? It's because we're talking about recessive disorders. So what would the genotype of a carrier look like? Well, this is where we write it a little bit differently. And the reason we write it differently is because now we have to include that X in there, just representing that we're talking about the sex chromosomes. So a carrier would look like this, and I just really want you to focus on the H's. We have a healthy normal gene, and we have a disease gene in the lowercase. And this would be what it would look like if the female was a carrier. Now, why don't I have males here? Well, that's because, as we already discussed, males are not going to be a carrier because if they have the disease gene, they're going to have the disease. Whereas in this case with a female, even though they have the disease gene, they have a healthy one that can overcome or overbear the unhealthy one. But with males, they don't have the two X's. So if they have any disease gene on their X chromosome, they have the condition. So an affected person then, we can look at both male and female. An affected female would have to inherit two of the disease genes, which you can see here, H and H on both X chromosomes. Whereas the male, we said, would only need to have it on the X chromosome and they would have the condition. So let's look at some specific examples of recurrence risk, carrier, normal, and males versus females. Remember, when we say normal, I mean that they don't have the condition and they're not a carrier. So in this example, we have a normal father. So you can see that normal H gene, and a carrier mother. So she has a normal and she has a disease gene. Let's see what the Punnett squared would look like. And it's just more appropriate for me to put the Y afterwards instead of at the start, in case you're wondering. Now, what's the recurrence risk? We want to know what are the chances that their child will have the condition. So we have to look at affected then and the diseased gene of H. So we're looking for this for female and this for males. So what we have is nothing there for female. And then for males, we do have one. So what that means is that there is a 1 of 2, and now we're saying 2 instead of 4 because remember we're looking at relative to males and females. So 1 of 2 chance that if they have a male child, it's going to have the condition. There's a 0% chance if they have a daughter or a female child that she's going to have the condition, right? Because we don't see what we would need to see, which is this, anywhere here. Now, what about the risk of being a carrier? Well, males don't carry, so that we know is going to be a 0% chance, or not relevant, I guess. But a carrier as a female would look like this. So there is a 1 in 2 chance that if they have a daughter, she will just be a carrier. So 50%. And remember, for males, it's really just not relevant because males cannot be carriers. Risk of being normal, we know normal looks like 2 H's, and we see 2 H's here. So risk of being normal as a female is going to be 1 of 2 because this one's not normal. That's a carrier. And this one here is normal. So for males, there's a 1 in 2 chance. So 1 in 2 for males and 1 in 2 for females. What about risk of male versus female of actually having the condition? Well, now it's finally relevant because we're talking about sex-linked disorders. So there's a greater chance of males having the disease. And we know that in this situation where there's a normal father and a carrier mother, it will not pass on to the daughter, but there's a 50% chance it will pass on to the son. So males. All right, now let's look at our next example. We have a father that has the disease, and this is what it would look like. Okay? We have a normal mother with 2 healthy genes represented by capital H. Let's fill out the Punnett squared. Now, what's the risk of male or female having the condition? Remember, the condition would have to look like this or like this. So let's see, dad's affected, mom is normal, not even a carrier. Let's look at female. Well, don't see anything for female. So if dad's affected with a condition and mom is normal, there is a 0% chance that it's going to be passed on to the female. For the male, there's also a 0% chance that it's going to be passed on. So isn't that interesting? If the dad is affected, but the mother is not, there is a 0% chance that they will pass this condition, the disease itself, to their offspring. So that's very reassuring for parents that if the father or the mother, or let's say the father, has a recessive disorder and it's present in him phenotypically, and he mates with someone who's normal and wonders, can I pass this disease on to my children? The answer is no. Now let's look at risk of being a carrier. So we know that a carrier is going to look like this, and that only females can be carriers. So it's 0 for males, but for females we have 1, 2. So there is a 2 out of 2 chance that the female will be a carrier. So that's a downside then. Even though they won't have the condition, the father will pass on that unhealthy gene, which will be carried by their daughter which is important for them to know for their offspring, should they have any. Risk of being normal, we need those capital H's, so we've only got a situation for the males, so the males have a 100% chance in being normal, and the females have a 0% chance in being normal. And remember, normal means that they don't have the disease and they're also not a carrier. They may phenotypically look normal, but they're not considered normal if they're a carrier in this context. So 2 out of 2 chance that the male will be completely normal, not a carrier, not having the condition, 0 out of 2 chance for the female. So a man that has a daughter will pass on the gene to both of his daughters and none of his sons. So equally not affected in this case, because when we look at the actual passing on and the recurrence risk, both are equally not affected. So we could say the risk for both is equal. And let's look at our final example now, where we have still an affected father, but now we have a carrier mother for that condition. Let's look at the Punnett square. Let's look at recurrence risk. So looking back to affected person, and this is what we're looking for. So for females, we can see this. For males, we can see this. So it's a 1 of 2 chance, or a 50% risk for both males and females to actually inherit the disease if their dad is affected, the father's affected, and the mother is a carrier. What's the risk of being just a carrier themselves? Well, we know that's only applicable to females, and we can see that there is a 1 in 2 chance that the female would be a carrier, and not relevant, of course, for males. Risk of being normal. This is what our normal one would look like. So we need to see capital H present on both X's for females, and we don't see that, but we see it for the male. So it's a 0 out of 2 chance that the female daughter would be normal, and there is a 1 in 2 chance that the male would be normal. And then males versus females, that brings us back to our recurrence risk. And the recurrence risk then is the same. Don't mind my spelling error there. Equally affected. So I would suggest at this point that you go back to that chart or that slide that I showed you earlier that talked about some of the rules of X-linked recessive disorders and apply it to what you've learned here. And then you can do the same thing after we've gone through now X-linked dominant disorders. So now we're talking still about that 23rd chromosome, the X-linked disease, because it's affecting the X chromosome. Y chromosome, remember, doesn't contain a whole lot of information, so most diseases of the sex chromosomes are X-linked. Because we're now talking about dominant disorders, that means that the disease gene is going to be represented by the capital letter. It's going to dominate over any healthy or normal genes. The genotype of a carrier would look like nothing, because there is no such thing. There's no such thing because with dominant disorders, the disease gene only needs to be found on one chromosome. So if you get the gene, then you are affected. Now, the affected person's genotype would look like this. We would either have, you can see here, the disease gene, which dominates over the healthy one. This, of course, is 2 disease genes, and that's for female. And then with males, if they have the disease gene, then they have the condition. Similarly, if they had a small h here, then they would not have the condition because it's just a healthy genotype. Okay, let's look at our situations here. We're going to look at an affected mother, and we're going to look at a normal father. Let's do our Punnett square. Let's look at recurrence risk. What is the chance that this condition will be passed on? So a mother with a disease says, I have this dominant disorder and I'm hoping it's not going to be passed on to my kids. What are the chances? So let's find that out for her. We know that this is what represents affected. And so for females, we see that this is a problem, but this one is okay. This is healthy. And for males, we can see that this is the problem. Again, that capital H is representing the disease gene, capital because it's dominant. So there's a 1 in 2 chance then, recurrence risk, or a 50% chance that if a mother has a condition but the father does not, and specifically sex-linked dominant, then they will pass it on 50% to 50% of their children, 50% chance that it will be passed on to their offspring. Risk of being a carrier, there's no such thing as carriers when we talk about dominant disorders, so that's not relevant. Risk of being normal, they would have to have those 2 H's, or 1 in the case of a male, so they have the healthy, and this one is healthy too because of 2 lowercase h's, representing the normal healthy gene. So 1 out of 2 for each of being normal. And then males versus females, you can tell right by looking at recurrence risk that it's an equal risk, equally affected. So now let's look at a flipped situation where it's the father now that's affected, but the mother's not. So just opposite to what we looked at. Let's fill in the Punnett square. And let's look at their recurrence risk. What is the chance that they'll have this condition? Come back up to affected person, so we know what those genotypes would look like. And when we look here, we see, okay, that's affected. We can see that's also affected. So there is a 2 out of 2 chance that if a mother is normal and the father's affected, he will pass that disease gene on to his daughters, and a 0% chance that it will be passed on to his sons. Not too favorable for females when dad has a X-linked dominant disorder. Risk of being a carrier now, no such thing. Risk of being normal, we know normal would be those small h's, so there's a 2 out of 2 chance for males being normal and a 0 out of 2 chance of the female being normal. That is not a carrier and not having the condition. And risk for this, we know that female is greater than males, and that's evident always looking back to that recurrence risk. Because that's another question that parents will sometimes have is what are the chances if it's a female? What are the chances if it's a male? And in our final example on the X-linked dominant disorders, we're going to look at a situation where the mother has a sex-linked dominant disorder. The father also has that sex-linked dominant disorder, which, by the way, is very rare that they would, because they would both be phenotypically, have the characteristics phenotypically, right? They'd have this physical presentation of the condition. So it is probably unlikely that they would mate with somebody with the exact same dominant disorder. But let's look at what it would appear as. So let's show everything out. And let's look at recurrence risk. So we're looking at affected. We're going to look at these 3 options. And what we can see here is that in females, that's considered affected. And so is that, because they both have that capital H representing the dominant disorder. So there's a 2 out of 2 chance that the female will get the condition if both the parents have it. But with the males, it's only a 1 of 2 chance. Risk of being a carrier, no such thing we know. Risk of being normal, we know it's 0 out of 2 for female. We've already clarified that, but with males, there's only 1 healthy genotype present here. And so it's 0% chance for female, and then 2 out of 2, 0% chance for a female being normal, but 1 of 2 for the male being normal, because this is the only normal genotype. And then males versus females. Unfortunately, females are not looking so good here. They are also more greatly affected than males when it comes to both parents being affected with an X-linked dominant disorder. So please now pause the video and go back to that rules list that I had on sex-linked dominant disorder and apply a little bit of what you've learned. So that's all on the Punnett square, and then we just have a little bit left to talk about what I promised I'd come back to, which is penetrance of a condition and expressivity. So penetrance of a trait is defined as the percentage of individuals with a specific genotype that also exhibit the expected phenotype. So what we're talking about here is somebody that has a specific genotype. What percentage of those people also present with the expected phenotype or physical characteristics? There are different types of penetrance that exist, and they are age-dependent penetrance, complete penetrance, and incomplete penetrance. Some conditions exhibit age-dependent penetrance. For example, Huntington's disease. The phenotype, so the physical characteristic with the signs and the symptoms, usually strikes in the we're talking about age, usually hits around the 30s or the 40s. And this is also true of familial breast cancer and polycystic kidney disease, which exhibit age-dependent penetrance. Complete penetrance means that all individuals with the genotype will express the disease. So all individuals that have the genotype will have the expected phenotype. Complete penetrance. Incomplete penetrance, on the other hand, means that individuals who have the disease-causing genotype may not exhibit the disease phenotype at all. So they may not have the physical characteristics, even though the genotype and associated disease can be transmitted to the next generation coming up. An example of incomplete penetrance is a condition called retinoblastoma. Retinoblastoma is an autosomal dominant disease that causes eye tumors in children. In the diagram on the right, the filled in red symbol are people with the genotype that have also expressed the phenotype. Retinoblastoma has an incomplete penetrance of 90%. This means that 90% of the people with the genotype for retinoblastoma will display the phenotype for retinoblastoma, which is development of the eye tumor. The remaining 10% of people appear normal, but they're considered obligate carriers of the gene, but they do not have the disease itself. So the black arrow showed here is an example of this, where the person has the genotype for this condition, but they did not exhibit the phenotype or the disease itself. But what's interesting is that when she has a daughter in this example, the genotype and phenotype are present. Now on to expressivity of a condition. Expressivity has to do with how the condition is being expressed. So you can have the gene for a disease, but it doesn't mean that that has that gene is going to have the exact same signs and symptoms of the disease. So it can be expressed to different extents, meaning that there's going to be variable phenotypes in how it physically presents. Even if many people have the genotype, they can look and present differently. So penetrance can be complete, as talked about on the previous slide, but the severity can vary. So what this will end up meaning is that you could have a parent that has a disease, but it's very mild, and they think, okay, well, if I pass it on to my kids, it will probably be mild too. Or it's so mild that they're not even aware that they have it. And so they can transmit then that gene to the child, as we've already learned about, who can then exhibit a severe expression of the disease. So it sometimes refers to being on a spectrum because it can be on opposite ends of the spectrum from a parent to a child. So the example that we're going to look at is called neurofibromatosis. Neurofibromatosis is a group of genetic disorders, so there's different types that can cause tumors to form on nerve tissue, really anywhere in the nervous system, so brain, spinal cord, and nerves. And so if somebody has the gene for neurofibromatosis, they could end up in the example A, where they have multiple dermal neurofibromas or tumors on their trunk, as you can see here. If you look closely, you can see them. Or they can have a large plexiform neurofibroma that just hangs off the right lower back. And these are just, of course, examples, but showing you the variation in expressivity of a condition. There's also variations with this condition not shown in these images here, where it can affect the spine and it can cause scoliosis. And one of the ways through school that I was taught to remember the association between neurofibromatosis and scoliosis was a song that I'm not going to sing, but it goes along the lines of neurofibromatosis, stop giving me scoliosis. Okay, so it is the different ways that the condition is expressed that we call expressivity. So someone can have these dark spots on their body and just think, oh, I've got a whole lot of moles, but not realize that they actually have the gene for neurofibromatosis. So in this case, they might not be aware, and then they can pass it on to their child who expresses the gene more severely than their parents. And as I'd mentioned already, we call this spectrum disorder, going from mild, maybe almost non-existent seemingly, to severe, but it can fall on different points along the spectrum. So why does this happen? Well, there's a lot of different thoughts on this. It could be because there's other genes that could affect the condition. It could be that there's other genes that modify the expression of the condition. It could be related to factors having to do with the environment. Maybe there's a slight mutation versus a more serious mutation that affects the expressivity. Lots of possible thoughts. Okay, so just at our last slide here, not a whole lot to go through, but on our final slide, we'll talk briefly about what diseases will affect us, and we're going to define a few terms. The first one is polygenic traits. Poly means many, genic refers to genes. These are traits that arise when you have multiple genes acting together. So in other words, genes can impact other genes. Multifactorial traits are variations in traits caused by genetics and environmental factors. So there's multiple factors, then we can say, by this name that can affect traits. So for example, the height of a person can be roughly determined by the height of the parents, but nutrition and access to healthcare can also affect this. IQ is another example, roughly determined by genetics, but learning environments, does the person have a tutor? Do they have really good nutrition? What was the intrauterine environment like for the baby? That can also have an impact on outcomes. There are diseases that are strongly affected by the environment and lifestyle. So influenza, and actually it's shown up here, influenza and measles. Exposure to other people increases your likelihood of getting it with very little genetic component other than maybe your immunity. Diabetes and heart disease, shown here, they're also impacted by the environment and lifestyle on this spectrum, but there is a genetic component, so it falls sort of right in the middle. So in other words, multiple variables affecting the development of these conditions. And then diseases such as cystic fibrosis and hemophilia A are listed here as having genetic causes. And you can see that on the spectrum. And so they're referred to as a term we've already used, single gene diseases. That's the end of today's lecture. Thank you so much for your time.